little bit of the story. So the story of this project is really a fascinating one. We're mostly interested in my research group in viruses you find in acidic, high-temperature environments, basically boiling acid. So viruses that can survive in boiling acid. So that was the real impetus for this project. We were looking, in fact, for some viruses that we thought would be in this particular Boiling Springs Lake, place in Nansen Volcanic National Park, but we couldn't find those viruses. So we took a step back and said, well, we're going to do a really high throughput, really massive sequencing approach to do this. So we took all of the appropriate fraction we could find that should have been about virus size, sequenced it like mad, got about 400,000 sequences out of it, and lo and behold, in those sequences, we found some sequences that looked like the viruses we were looking for. But the real surprise was we found some sequences of viruses that we weren't looking for at all. And so this is the impetus for the publication, which we just got the prize for. And that was, we found sequences in our DNA sequences that were similar to sequences from RNA viruses. And this was odd. At first, we didn't believe it. In fact, I didn't believe it at all. I told the graduate student to go back and do it again. So then he was very carefully showed me that these really were sequences was present in the DNA. They're actually on the same piece of DNA as a DNA protein, DNA virus, known DNA virus protein, that we then were able to go back, show in the environment it was there. And moreover, we were able to then show with these sequences that it wasn't just in this high temperature acidic environment. It's actually spread throughout the whole world. And no one had actually really noticed it, or they'd ignored it in the past. And so that's really the big breakthrough that we made. Couple of reasons that we made this breakthrough and nobody else did. One, we're looking at a very low diversity environment, high temperature acidic environment. So very few sequences in there, very few viruses. So we're able to put these two pieces together, two pieces of the jigsaw puzzle. And the other aspect of things, I like to think it's really kind of like Louis Pasteur and dans le champ de l'observation, la chance ne favorise que les esprits préparés, generally translated as chance favors the prepared mind. So we were ready to see this, we found it, and then went forward and were able to publish it. Now, the real aspects of this discovery, what does this mean? I think it means, and this was in fact one of the reviewers in Biology Direct, which is there, it's published with our paper, which says this really is a major insight into how viruses are evolving right now. What are the new viruses? What are the new viruses happening? How are these things coming about? And being able to take or literally steal an RNA from an RNA virus and put it into a DNA virus, that's amazing and just completely unheard of when we found it was happening. And now the big question is, how is it happening? How frequently is it happening? And so that's the real area of research that we're moving into right now.
That's lovely. That's really, really nice. Yeah. Okay.